Okay, welcome everyone. I'm Joon Lee from Cellatoz Therapeutics. I'm a senior EVP. As you can tell from our name, Cell A to Z, we're a cell therapy company based in Seoul, Korea. I'll explain briefly about us and some of the programs within the company. So we were founded in August of 2017, a little over 4 years ago. We are a team of 48, mostly from major biotech companies within Korea. And we've raised cumulative total of about $40 million, both in dilutive and non-dilutive funding within Korea. And we have 4 proprietary cells, 3 of which are in preclinical stage. We utilize functional cells that are derived either from primary stem cells or pluripotent stem cells. So the advantages are clear MOA and real regeneration and changes in microenvironment. And we have capabilities in production and quality control of cells, maintaining consistent efficacy, with strong CMC, and we're using allogenic frozen formulation and thus easier mass production. This is a quick snapshot of our CGMP facility. It's about 15,000 square feet in size. We've acquired 2 licenses from Korean government this year, and we're in line for an audit by Ph.D. of Japan. This will suffice most of our needs, but if you need a partner or CDMO in Asia, we can be of service to you. So onto our proprietary cells. First one is a neuronal regeneration promoting cell. It's derived from tonsillar mesenchymal stem cell, and as the name suggests, it promotes regeneration of nerves. And so we're targeting Charcot-Marie-Tooth type 1A disease as the initial indication. And if successful, we plan to expand to other peripheral neuropathies and even to spinal cord injury. The second one I want to introduce is musculoskeletal stem cell. It's an IP-protected cell that can differentiate into various muscular skeletal system. And these are derived from embryonic stem cell and iPSCs. And are initially targeting osteoarthritis of knee, and we will expand to other musculoskeletal disease, including complex fractures. The third one is a natural killer cell with a high expression of CD16, and I think it's suitable for repeat dosing, rebalancing the immune system, so changing the immune profile of a patient to combat cancer. And with high expression of CD16, we think we can do combination therapy with existing cancer drugs, taking advantage of ADCC. And we're targeting solid tumor. The initial indication we're looking into is glioblastoma. So NRPC, codenamed CLZ-2002, we will be filing an IND in the 4th quarter of this year, both in Korea and in the US. And we'll be initiating phase 1/2A clinical trial as early as 2nd quarter of 2022. MSSC, codenamed CLZ-1001, targeting OA of knee, will be submitting in our IND in the 2nd quarter of next year. And our NK program, which is only autologous program that we have, will be submitting an IND in the 3rd quarter of next year. So on to our most advanced program, NRPC. Using proprietary method from tonsillar mesenchymal stem cells, we were able to differentiate a neuronal regeneration promoting cell, in short NRPC. These cells have unique properties of assisting in neuronal regeneration and myelination, and so has similar functions as Schwann cells. So we sometimes call them Schwann cell-like cells. And the characteristics are, in morphology-wise, they're bipolar or tripolar in spindle-shaped. In our neurite outgrowth assay, we observed a neurotrophic effect, increasing the total number and length of neurites. And in myelination assay, when co-cultured with dorsal root ganglia, we observed formation of myelin sheath. So with that in mind, I'll divert a little bit to and give you information on the Charcot-Marie-Tooth disease. It's a rare disease without a cure at this time, and it's quite well known in Korea because 2 of the richest families have been afflicted by this disease, and mainly due to hereditary reasons, a person will gradually lose motor abilities and sensory impairment sets in, and then muscle loss comes in. And over 90+ genes have been implicated as a cause of various types of CMT. And as you can see in the photos, the disease starts from feet and hands and moves up the limb and is quite debilitating. And about 1 in 2,500 people are afflicted by this disease. That translates to about 20,000 in Korea and over 1.5 million patients worldwide. We are pursuing orphan drug designation in the U.S., and 4 major types of CMT actually covers 90% of the patient population. And CMT1A, which is the most prevalent form, covers 50% of the patient base. And fast development is possible through RMAT in the US and Sakigake designation in Japan. A little bit more on type 1A. If you see the photo on the left, it's the exon where the Schwann cells are sheaths that covers the exons. But when PMP22 genes are overexpressed in a person through protein missorting, Schwann cell becomes defective and demyelination sets in and eventually leads to muscle atrophy. So in developing a therapy for this disease, restoring Schwann cell defect and reversing the muscle atrophy are key factors to consider. And our goal here is to use NRPC's Schwann cell-like properties, which to restore the damaged nerves through remyelination and slow down or reverse muscle atrophy. So some of the efficacy test results, we tried them on two different mouse models. This is Trembler J mouse model. This is the wild type, the normal mouse. This is a Trembler J mouse. It barely moves. And this is a Trembler J with a single dose of NRPC injection after 12 weeks. You can see it behaves more like the wild type, especially being able to crawl up the wall, which is a significant mouse behavior, meaning that the muscle strength is being regained. This is the rotorod test results of the Trembler J mouse. Focus on the graph on the right. The white bar is the Trembler J, and the black bar is the NRPC-injected Trembler J. By week 12, you can see that the mouse hangs onto the rod for over a minute, again implying that the muscle strength has increased. So this is the Immunostain picture. Focus on the red box. Image D is the wild type, where you see the red exons enveloped by green in a very uniform way. The image I is the Trembler J. There is absolutely no myelination, completely demyelinated. And then image N is the Trembler J with NRPC injected. And in week 12, and you can begin to see remyelination, like green color enveloping the red, which are the exons. So we believe through regeneration of the sciatic nerves by remyelination, the restoration of the muscles and muscle strength and increased mobility actually happens. And this is C22 mouse model results, which are similar. C22 mouse model mimics CMT1A in humans in a severe way. Image on the right, on your left, it's the normal mouse where you see nice green donuts covering the red exons. The image in the center is the C22 mouse where there is no myelination. And image on the right is the C22 mouse. With 2 injections of NRPC, 1 on each thigh. And you can see green color enveloping the red here and there. That's the remyelination of the damaged nerves. This is the rotorod test result of the C22 mouse model, which is quite drastic. Image on the right, a video on the right is the C22 mouse. It just falls off the rod within 11 seconds or less. But the video on the right, the NRPC injected mouse actually stays on for more than 3 minutes, and the wild type actually falls off at about 5 minutes. So there is a drastic improvement in the muscle strength. And this is a preliminary design overview of our phase 1/2A clinical trial. We plan to enroll mild to moderate stage CMT1A patients, and the NRPCs will be administered via intramuscular injection in the lower legs, the 3 muscles, gastrocnemius, peroneus longus, and the tibialis anterior muscles. And we'll have 2 arms, and each arm will have 2 groups. Arm 1 will have 6 injections, meaning 3 injections on each leg. Arm 2 will have 12 injections, meaning 6 injections per leg. And in dose escalation, per injection, we're looking at 1 million cells and increasing to 2 million cells per injection or per site. So switching on to our musculoskeletal stem cells. From human embryonic stem cells through proprietary methods, we were able to culture MSSC. This cell is similar to your hematopoietic stem cell for blood, where that it can differentiate into various musculoskeletal systems in the body. And we filed our first patent in 2017, and when the patents were approved in US, Europe, Korea, Japan in 2020. An interesting thing was when we first filed our patent in 2017, no one really believed such cell actually existed until 2018 when Dr. Longacre's group from Stanford Medical School discovered that there was a human skeletal stem cell that existed in the bone marrow, which were quite similar in function. But obviously it had different biomarkers than our cells, so our cells are patented and can be mass produced because these are artificially made. So some efficacy results. In a mouse Foley model, we deliberately removed portions of the bones so that no natural healing could occur, as you can see with the photos without MSSC. But the below photos, you begin to see the formation of the bones when MSSCs are injected. And the interesting thing is that these bones are actually of human origin. And photo on the right is the OA mouse model where we injected MSSC into the mouse joint. The green color is a cartilage formed as a result, which implies that our cells, when it's injected, has regenerated into cartilage. So it means that real regeneration has actually happened. So we're considering, as our first indication, OA of knee, more specifically grade 2 and 3 through direct injection rather than surgery, because the environment in the knee actually ensures our cells would only differentiate into cartilage and not into other form of musculoskeletal system. So lastly, natural killer cells. We believe in balancing the immune system to change the profile of the immune profile of the patient to combat cancer. So in order to rebalance the immune system, we believe that multiple infusion of autologous NK cells are needed. That's why we are mass culturing them, both in live and frozen formulation for multiple infusion. And our cells have a unique characteristic of showing high expression of CD16, which will enable us to do combination therapy with drugs like Herceptin. And in our preliminary studies, showed that increased ADCC as the dose of Herceptin increased. So upcoming milestones and next steps. Next year, we'll plan on initiating at least 3 clinical trials. And we plan on establishing a North American entity to handle activities outside Asia, such as clinical trials in U.S. and further financing for developments in U.S. or EU. And we plan to build out more partnerships to collaborate on the development of MSSCs. Well, thank you for your time. That concludes my talk. And I have about 90 seconds. And if you have any questions, I can answer them later as well. Okay, so if you have any questions, come to me in the back. Thank you.